Hello, Ken.
Oh, hi. Nice
How are
to
you?
see you. Okay.
Okay, how are
Fine,
you?
thank you. Yeah. Oh, it's
Hello,
also
Dr.
good.
Sato.
Yeah.
Hi, thank you. How are
Nice
you
to see
doing?
you.
Yeah.
Indeed, my great pleasure and honor to welcome you all to participate in this very first webinar of ASIATOX. Because of the COVID-19 pandemic, we are not able to meet each other as frequently as they used to be. So to overcome this pathogen-created barrier, we try to share information and exchange. Right? So now let us welcome Dean of Pharmacy, Kaohsiung Medical University, Professor Huang Yaobin, to give a few words for welcome remarks.
Good afternoon, everyone. It is so nice to join this prestigious online conference with all of you during the COVID-19 pandemic. I'm Yaobin Huang, and the Dean of the College of Pharmacy from Kaohsiung Medical University. Before the beginning of the meeting, let's welcome our friend from Japan and Taiwan. And I would also like to take this opportunity to thank Professor Lee, who is the president of ASIATOX and who makes this meeting possible. As we all know, the goal of this conference is to share the expertise of the ASIATOX Society members and the extended network. After today's meeting, I believe every member of this community will have better understanding about the topics discussed today and form a better friendship with each other. Now, let's first welcome Dr. Tatsuo Sato, who is the advisor of ASIATOX, and he will share his opinion about past and future perspectives of ASIATOX. Next, we have Dr. Jun Kano, who is the president of JSOT, and he will talk about comprehensive analysis of the gene expression networks in toxicology. After that, Dr. Yasumitsu Ogara, who is a professor at Chiba University, will address biological and toxicological significance of tellurium. Finally, Dr. Yingran Wang, who is the president of the TSTA and also the alumni of KMU, will discuss the possible role of autophagy in nanomaterial toxicity and safety assessment. With the sharing from these professional and outstanding scholars, I know we can learn so much from them today. This is only the beginning of the international academic exchange, and there will be more activities like this in the future. I hope you all enjoy the rest of the meeting. Thank you very much. Thank you.
Thank you, Dean Huang, for the welcome remarks. And my appreciation also extends to President Wang Yingran and many friends in the Toxicology Society of Taiwan who have organized this seminar, and also to Dr. Sato, Dr. Kano, Dr. Ogura, and to Dr. Ning Yingqi. All of them proactively help organize this seminar. Without their kind help, this seminar would not be possible. Today we have friends from 6 societies. In addition to Japanese Society of Toxicology, who help a lot for this very first seminar, and TSTA, as I just mentioned, we have 4 other societies. These include Chinese Society of Toxicology. I'm very pleased that President Zhou Pingkun, and Dr. Fu Lijie, who is also the immediate past president of ASIATOX, and some other friends I have known for years, such as Guan Hua, Xie Yaohua, Li Hua, Wu Xiangjun, Chen Chunying, Wang Chunjun, Tai Yi, Wang Ruixue, and many others I have known for years. And also Malaysian Society of Toxicology, Dr. Rosani Abdullah and Mary Koo, and also Toxicology Society of Singapore, Dr. Ponampram, as well as Thai Society of Toxicology. I know Dr. Songsak for many years, who also helped me a lot, and Dr. Shanifun Butri, I hope I pronounced her name correctly. So with all your participation, I sincerely welcome you all, my friends, and hope the success of this first webinar will bring forth the continuation of subsequent webinars to the ASIATOX 2023 Congress held in Tainan. Today, we have 4 distinguished speakers, as mentioned by Professor Huang. So now let's begin with the first session. This session will be given by Dr. Tetsuo Sato, who is now advisor of ASIATOX. Can I have next slide, please? Dr. Sato, in addition to the advisor to our ASIATOX, he also Professor Emeritus, Chiba University. He graduated from Toyama University with a BA degree in pharmacology. And then he obtained his master's and PhD degree from University of Hokkaido in pharmacology and toxicology. During his 40 years career, he has been in many important positions, such as Vice President of the International Union of Toxicology, ASIATOX, Advisor of Asian Society of Toxicology, ASIATOX, Emeritus Members of the Society of Toxicology, USA, Emeritus Member of the Academy of Toxicology Sciences, ATS, USA. He was once served as Associate Professor in the Toxicology Laboratory, University of Chicago. In the past 4 years, he published over 400 peer-reviewed papers and book chapters. I admire you a lot. I think I never reached 400, such a few volumes, and contributing to the scientific world. So without further delay, I would like to introduce Dr. Sato for the topic ASIATOX Past and Future Perspectives. Let's welcome Dr. Sato.
Good afternoon, ladies and gentlemen. It's my great pleasure and honor to have this opportunity to talk about Asian Society of Toxicology. Also, I sincerely appreciate Dr. Lee to invite me to this wonderful seminar. The topic of my talk this afternoon is ASIATOX Past and Future Perspectives. This time I'm talking about history of the Asian Society Society of Toxicology. Second, background of the ASIATOX formation and the society's meetings and events. Third, general objectives of ASIATOX. Fourth, International Union of Toxicology. And finally, I'm talking about conclusions and future perspectives. Before the ASIATOX formation, we had the Japan-Korea joint meeting on toxicology every year. In 1994, this joint meeting was expanded to other Asian countries to form the ASIATOX. The charter member societies include the societies of toxicology in Japan, Korea, China, Thailand, and Taiwan. Later, the Iranian Society of Toxicology, the Toxicology Society Singapore, and the Malaysian Society of Toxicology joined the ASIATOX in 2012, 2013, and 2018, respectively. And I'm hoping member societies will be increased in future. As of January 20, 2018, ASIATOX boasted more than 3,000 members from a variety of industry, academia, and regulatory organizations. ASIATOX joined the ASIATOX membership on July 2nd, 1995, and 300 members among ASIATOX member societies were registered to the ASIATOX membership. This slide shows the founding executive committee members of the ASIATOX from 1994 to 97. President Dr. Tomoji Yanagida, he was the expert of the drug addiction studies. And as a president, he showed excellent leadership. And first vice president, Dr. Sandai Park from Korea. Second vice president from Dr. Jianlin Zhou from China. And the Secretary General, myself. Treasurer, Dr. Kim Fan-Yang. He was very active as the treasurer. And we had 5 councillors. Dr. Hitoshi Endo from Japan, Dr. Yansun Lee from Korea, Dr. Ilman Chang from Korea, Dr. Minda Wang from China, and Dr. Parap Sihasini. Dr. Endo is still active to manage his own company. And Dr. Sonsak, as you well know, he is continuing his outstanding performance in many fields, not only in Thailand, but also worldwide basis. At the General Assembly of the ASIATOX 18 in Thailand, 2018, the bylaws of the executive committee members were amended. The names of first vice president and second vice president were changed to President-elect and just Vice President, respectively. In addition, immediately past President was newly added to the Executive Committee members. This slide shows the general objectives, recognizing the value of multinational organization. ASIATOX will work toward the following objectives. First, serve as the scientific voice of toxicology in Asia under the ASIATOX umbrella. Second, provide leadership and a forum for promoting scientific cooperation and information change related to toxicology in Asia. Third, facilitate platforms for discussion among regulators and academia regarding toxicological issues. Fourth, improve dialogue with member societies and other toxicology societies worldwide. Finally, address leadership as a scientific organization based on the state-of-the-art knowledge in toxicological sciences. ASIATOX congresses has been held every 3 years. First Congress was held in Yokohama, Japan in 1997. Second Congress was held in Jeju, Korea, 2000. Third Congress was held in Bangkok and Chiang Mai, Thailand, 2004. And next year we have the ASIATOX 10th Congress in Tainan, Taiwan, 2023. And at the future congresses, ASIATOX 11 will be held in Malaysia, 2026. And 3 years later, ASIATOX 12 will be held in Japan in 2029. I'm talking about International Union of Toxicology, ASIATOX. ASIATOX was founded in 1980, and it is the voice of toxicology on the global stage. ASIATOX has now 63 national and regional society members as of June 2022, representing some 25,000 toxicologists worldwide. Other regional societies of ASIATOX, we have 3 toxicology societies: Asian Society of Toxicology, ASIATOX, and the Federation of European Toxicologists and European Societies of Toxicology, EuroTox, and the Latin American Society of Toxicology, ALATOX. International Congress of Toxicology had been held every 3 years. First Congress was held in Toronto, Canada, 1977, and in the Asian region. Fourth Congress was held in Tokyo, Japan in 1986, and the 13th Congress was held Seoul, Korea, 2013. And this year, 6th Congress will be held in the Netherlands. This slide shows the ASIATOX Executive Committee members representing from Asia. Dr. Fuminori Sakai from Japan as the second vice president, Dr. Fukuda, second vice president, and myself, and Dr. Kurokawa from Japan, second vice president, and Dr. Inoue, second vice president, and Dr. Kanno, Japan, was elected as second vice president in 2009, and Dr. Kanno was elected president-elect in 2013, and he was elected as president in 2016 for 3 years. And at the moment, we have 3 directors of ASIATOX from Asian region. Dr. Kumagai from Japan, and Dr. Li Jie-fu from China, and Dr. Salman Hassan from Malaysia. This slide shows the conclusions and the future perspectives. As a regional voice of toxicology, it is vital that ASIATOX continue extending knowledge of toxicological issues throughout Asia by the following issues. First, providing leadership as a worldwide scientific organization that objectively addresses global issues involving the toxicological sciences. Second, broadening the geographical base of toxicology as a discipline and profession to all countries of the world. And third, pursuing capacity building in toxicology, particularly in newly industrialized countries, and utilizing its global perspectives and network to enhance both toxicology education and the career development of young toxicologists. At the end of my talk this afternoon, I'm very much happy to meet old friends and new friends in Asia. I'm hoping that we will be able to meet at the ASIATOX 10 in Tainan next year. Thank you for your attention.
Thank you so much, Dr. Sato, for such a comprehensive talk on the history of ASIATOX and a suggestion for the future direction. Especially you mentioned China many times, so that all the audience should have known that we are going to have the ASIATOX Congress next year in Thailand. Thank you. I remember a Chinese saying, old saying by Emperor Tang Taizong. He once said, 以史为镜，可以明得失, which means with history as the mirror, the right and wrong, we can learn from the experience. So in your experience, could you let us know that what we have done is good enough and what we have done is not good enough so that we can improve ourselves to go forward? Your microphone has not been turned on.
質問が 今まで
Yeah, I can hear you now.
Okay, I'm very sorry to this.
It's okay.
Yes, I think the ASIATOX is now growing, and I'm just hoping the number of member societies will be increasing in Asian region. So the current member societies are very much doing best contribution to ASIATOX.
Mhm.
So I really hope to extend our member societies to Asian countries. That is my hope.
Okay, thank you. The podium is open to all the participants. If anyone would like to raise question to Dr. Sato, we still have some time, especially for the previous presidents of ASIATOX. You probably have some question to ask or to suggest. Anyone?
I have a question. May
Yes,
I?
please.
Okay. Santoha, thanks for your wonderful speak. And I have a concern that if we want to push our toxicology society to all scientists across the world, but at the current station, it is greatly influenced by the pandemic such as COVID-19. So if we face this change, how to deal with these things to connect all the scientists across the world working together in the toxicology? Thank you, Setoha.
Yes, actually, it's very difficult to answer correctly. So because the pandemic happens in the when and where happens in the world. So I have no good idea to make to your questions. So I think the pandemic is very, very, dangerous. When and where happens, which country pandemic happens. So it's very much was difficult to say exact answer. I have no good answers to your question.
Well, not an answer to Dr. Huang. Dr. Huang Ruixue, who is from China. We met in 2018 in Pattaya, right?
Yeah.
Yes.
My opinion about your question is the world is getting smaller and smaller, and because we have now the information exchange quicker than it used to be, so that probably we have a problem at one country, one place, but with a good monitoring system, then we can detect the problem and send out the information to other countries or other places so that we can inform the other places so that the danger of the chemicals or the pathogens will not spread as quickly as it was. So that probably one way to do. And the other thing is, as a toxicologist, we probably can work together because we have different training background and different specialties. So once we find out a chemical or pathogen that probably dangerous to our health, then we can summon experts from different fields so that we can work together. That's actually my second question to Dr. Sato as well. In your conclusion, you mentioned that as a regional voice of toxicology, it's vital that ASIATOX continue extending knowledge of toxicology issues throughout Asia. So probably we, through the ASIATOX, we had a platform and we have a system. The networking probably can help us to work together so that whenever we have a problem in some areas, then we can work together to solve the problems. And that's the way we should have the ASIATOX. So that's just my thought. Probably not the answer, but just something complementary to Dr. Huang and Dr. Sato's discussions.
I have no particular answer to your questions. Okay.
Good afternoon, everyone.
Yes.
Dr. Lee
Yes.
and Dr. Songsak.
Dr. Songsak, nice to see you.
Yes.
How are you?
Very good. How are you? Well, according to the system that we have in Thailand, we have to watch the situation in various countries around the world very closely and have a system that to organize or to control the pandemic in Thailand very strictly by using the law. However, for ASIATOX, I believe that if we have what we call the alert system among our member societies, it would be good, not only for the pandemic of the pathogens or the virus, but also the alert system for the chemical toxicology as well. I think that if we set up the alert system, it might help in preventing or management of the problems quickly, particularly among our member countries and other interested countries in Asia, I believe. Yes, this is my thought and probably a proposal for establishment of the alert system to the present president of the ASIATOX. We can discuss that in the council meeting and executive meeting in Taiwan. Yeah.
Thank you, Dr. Songsak. I think that's a very good suggestion. With a warning or monitoring system set up in each society in every country. So whenever we have a problem in one area, and then we can detect and bring up to share the information and then try to solve the problems. let's bring it up next year in the ASIATOX 2023 in Tainan. I think that's a good issue.
Yeah, I think that's a very good suggestion, a good idea, to hold some symposium, something in Tainan meeting.
So Dr. Songsak, are you prepared to give a talk next time? Since we are now in the first session, I contacted you previously, and you said you are quite busy. So
Yeah.
I've been expecting you.
Yes, because at the moment I'm quite busy with the meeting in the regulatory committees, various committee in Thailand.
Yeah,
But the
okay.
next session, probably, yes.
Okay, great. Nice to see you again, though.
Yes, nice to you all.
Since we have 1 or 2 minutes, may I suggest that we can have a group photo now? So please turn on your visual aid now, so that we can have the group photo for all the participants. That's great. That's good to see the faces of everyone happily.
Professor Zhen, could you see me?
Can you hear me?
Yeah. Could you see me?
Yeah, sure.
Okay.
Now I can see, Dr. Zhou Pingkun, President of CST, and my friend from Iran, right? Hello.
How are you doing? It's good to see you.
Good to see you.
This is Omics Observatory.
So I also hope that you can give a talk the next time.
Hope so. I'll talk to my colleagues at Iran Talks and
Okay,
inform you.
Great. So everyone turn on the visual aid. Ready? 你那边可以吗? Okay. So cheers. One more. 1, 2, 3. Okay. 可以吧? Okay. Okay, thank you. And let's thank again for the group photo and thank you for the contribution from Dr. Sato. And let's move on to next session. The next session is chaired by Dr. Wang Yingran. Can I have the slide, please? The second topic, comprehensive analysis of the gene expression networks in toxicology, single versus repeated dosing, and organ-organ interaction. This topic is given by Dr. Jun Kano. He's currently the president of the Japanese Society of Toxicology. And the moderator is Dr. Wang Yinran, president of TSTA, Toxicology Society of Taiwan. Can I have next slide? Next slide. The moderator, Wang Yingran, Professor Wang Yingran, 上一张. He's now our president, TSTA, He graduated from Kaohsiung Medical University, the same university as I attended. And then he obtained his master and PhD degree from National Taiwan University in biochemistry and molecular biology. He's now distinguished professor of the Department of Environmental and Occupational Health, College of Medicine, National Cheng Kung University. He's going to hold the Congress because next year we are going to have the meeting in Tainan, and he will be the host, one of the hosts. Let's welcome Dr. Wang Yongnan to chair the session.
Thank you, Professor Lee. I'm going to introduce you our next speaker. Please show me the next slide, please. So our next speaker is Professor Jin Kanong. Jin Kanong is right now serve as the visiting researcher in both the Division of Cellular and Molecular Toxicology and also in the Division of Risk Assessment in the National Institute of Health Science, NIHS. And he's also right now the director, the medical director of the pathology department in Nissan Tamagawa hospitals. And Professor Jin Kanong got his MD PhD degree in Tokyo Medical and Dental University. And two of the most impressive experience, he was the director of Japan Bioassay Research Center in Japan Organization of Occupational and Health and Safety. And the other one is also the director of the Division of Cellular and Molecular Toxicology in the NIHS. And Professor Jin Kanong is the immediate past president of the ASIATOX and also the current president of the JSOT, the Japan Society of Toxicology. And his research interests include the experimental pathology and also the human pathology. Based on those impressive background, we can expect a wonderful talk from Professor Jin Kanong. So please welcome Jin Kanong. We can, very sure it's a very impressive talk. Kanong, please.
Thank you very much for your kind introduction, and it's a great honor to be one of the speaker of the first meeting. May I upload my slide from my side? Can you allow me to do so? Okay, thank you. Okay. Can you see my slide?
Yes.
Okay, then is it a slideshow right now?
Yes, we can see that. Wonderful.
Okay, so I have a multi-monitor and I have to turn around to look at my slides. So I looked, yeah, I speak with this position. Thank you so much. I've been very lucky to allow running a long-standing toxicogenomics project at the National Institute of Health Sciences. It was in Tokyo and now moved few years ago to Kawasaki, where it's next to the Haneda Airport right now. And this study is in collaboration mainly with this Systems Biology Institute. And we started this project nearly 20 years ago, and the first motivation was to avoid the thalidomide story-like issue again. The thalidomide, it was hard to detect its teratogenic effect by rodents, and it happened in humans first. That was the tragedy, and yet we had only a method for majority of the chemical toxicity to find out by experimental animals. And currently, many countries still uses this safety factor or the uncertainty factor to somehow use this rodent data to humans for regulating chemicals. But still, we are handling the organism or the living experimental animals and humans as a black box, and we thought we should look into the black box as carefully as possible to avoid this species difference and a very complex toxicity like thalidomide. And we wanted to look into the black box, but as you know, toxicology is we don't know the target yet. Pharmacology, you know the target first so that you can select where you want to look at. But for the toxicology, it's hard to find where. So we thought we should look comprehensively. And so if you think the black box as a body, a chemical as an input and symptoms as an output, we thought we should look into the network within the black box. And when we started this project, there was only one method available, practically available to look comprehensively was a DNA microarray chip looking for messenger RNA. We thought about protein, but it was so difficult to look comprehensively about protein, especially something like phosphorylation and so on. So we decided to keep our level of study at messenger RNA level. And yet we thought we should pile up a lot of data. We decided to make a normalization technique. We wrote a paper in 2006. And in short, what we did was to spike the sample by bacterial RNA so that we can measure how many copies of our messenger RNA are in the homogenate per one cell. So we count the cell number in the homogenate by measuring the genomic DNA, and now we know how many cells are in this homogenate so that we add spike RNA from outside. And now we had a method so that we can count messenger RNA copy number per one cell in average. Not really looking into single cell. We are looking as a whole organ or the sample, but in average, one copy, 2 copy, 10 copies of messenger RNA per cell. And we decided to first look into the very initial response of the animal organs using mouse. The reason why we took mouse was because we wanted to use knockout mouse, gene knockout mouse in the future. We thought rat was very difficult at that time to have various knockout rats. So we decided to stick to mouse for this project. And we wanted to look into time course and dose response at the same time. So we prepared this protocol. So we use 48 animals, 16 animals per group. And so we dose vehicle control, low dose, middle dose, and high dose to 16 animals, and then sample every 3 after giving vehicle low, middle, high dose after 2 hours, 4 hours, 8 hours, and 24 hours sequentially, and sample the liver first, and we run this Affinitrix gene chip. And again, we normalize the data against the cell number so that the data is, the data can be drawn into this 3-dimensional graph, like on the right side, and but before that, you toxicologists should be very interested in how we selected the dose. This is a very important part of our project. The top dose was selected that maximum dose without microscopic and microscopic changes in 24 hours. That means there is no overt toxicity. We avoided cell necrosis in liver in 24 hours because if you have a necrotic figure in histology, you have a microarray data which is full of necrotic response or the strong, how do you say that, cytokine release and so on. So we avoided such a strong effect, but we kept this dose as low as possible to look into the signal changes. And for the hormones, we selected ED 70 or 80 equivalent dose. And the dose ratio, we selected root 10, which is like 10 milligram, 3 milligram, 1 milligram, or 27 to such kind of nearly 3 dose ratios. And we find that the data looks like this. If you have a very clear response, it's like on the left side, which has a very beautiful dose response at 24 hours in this case of a CYP1B1. On the right-hand side, this chemical didn't induce anything. And if you look at the vertical axis, it's the maximum is 1 copy per cell. And the Affymetrix gene chip sensitivity is around 1 copy per cell, so that it's quite noisy. And we simply see that this is noise and doesn't mean anything biologically. And if you look carefully to the shape of some this three-dimensional surface graph, we call it surface graph. But you can see V, L, M, and L on the left-hand side, and V means vehicle control. And if you follow the timeline, 2 hour, 4 hour, 8 hour, the vehicle shows a spike in 8 hour, and this is a circadian change. And then if you look at the 4 hour green line, it shows that there's a dose response at 4 hours. And this yellow asterisk means that it is statistically significant by t-test. And good thing about this expression of a 3D graph is that you find out many genes which respond like this. This is from one study and one chemical. And now you find out this highlighted 3 genes shows very similar response. So that we thought this kind of a clustering by the shape of the surface graph, 3D graph, we can assume that these listed clustered gene should be or can be under same signal regulation or the similar regulation, which means that they may have a common upstream regulators. And yet at the same time, we find that there is a strong circadian changes in all organs. So that this is one of the take-home message to you all, that if you use animal for the messenger RNA, you have to keep your animal facility light and dark cycle very precisely, because the expression can change 10 times by only timing of when you sample. In this case, this DBP circadian genes, the liver, the pink colored shape, which shows that the wave is going from 10 copy or 7 copy at 2 hours to 70 copies at 8 hours, so that you instantly know that if you mix your animal and sample at 10 o'clock, 11 o'clock, 12 o'clock, and afternoon, simply by the date, time you sacrifice the animal, the data can change from 7 to 70. And if you average out that data, you may get nothing. So if you handle the animal, and if you are looking at messenger RNA, you have to be very careful about, again, how you keep the animal in the light dark cycle of your animal facility, when to dose the animal, and when to sample your animal. And it is not only about gene chip. It is only about RNA-seq or next generation sequencing. If you measure the messenger RNA from the animal sample, you have to be very careful. This type of a change is not only circadian. There are so many, like 5,000 genes in liver, and the shape of the curve is, in this case, it's going up at 8 hour, but there are many which shows opposite. 8 hour at the lowest and come back at 24 hours. So please be careful about that. But anyway, so again, we thought that by the shape of the surface, we thought we could cluster, and we could assume that this clustered gene list is possibly under same or common upstream regulation. So that we thought from one chemical, at least we can cluster the gene according to time and shape, because the peak, we can see the peak from 2, 4, 8, and 24 hours. We can cluster by that and then by more precise shape. But anyway, we thought we could cluster time-wise, which is the reflection of how the signal goes through time. And so we wanted to look into the network which is governing this time course and those response. We thought we could somehow extract out the gene list which has some meaning with a common upstream regulators. And we thought one chemical data is not enough. We thought two is better because we can look into the intersection of the ripple. You can see this cartoon. One chemical has a kind of a scalar type, not vector type data in terms of a clustering, like 2 hour, 4 hour, 8 hours. But if you have two chemicals, and if you find same gene list at the same certain group by comparing two data, chemical A data and chemical B data, we thought we could finally get some idea of what is happening by just looking into our data, not referring to the published data. In other words, unsupervised way. And now we are collecting more than 150 chemical data using the same protocol. We thought this ripple type idea can be extended from the flat water to a globe floating in the universe. And so this, please consider this sphere as a liquid sphere, and we are challenging this sphere with multiple chemicals making ripples, and we look into the intersection of multiple chemicals. And we did a little bit with a computer system using a program written by our colleague, Dr. Aisaki. And we have a gene list roughly sorted out by software so that, and if you click a chemical here, you can see a red marking estragol, and then the software gives you which has an intersection with clofibrilate and DEHP, diethyl hexylphthalate. And you can go into further, and if you look at the intersection between this estragol and clofibrilate at 2 hours, we instantly find there's a gene PDK4. I think PDK4 is very famous, that it is under regulation of a PPAR alpha. And you may see that this 3D graph of estragol and clofibrilate of this PDK4 looks very similar. So that one thing is that our dose selection was good, and then we find that these two chemical is triggering the same, probably the same upstream regulation in the mouse liver. And so indeed, this comes out later, but this gene is of course under-regulated by PPAR alpha. And estragol is the ligand for PPAR alpha, and clofibrate is the very famous PPAR alpha ligand. Now, quickly jump into the repeated dosing. This is the main topic today. And the talk until now was single exposure follow-up study up to 24 hours, how the signal goes through the liver, triggered by the chemical. Now we wanted to know what's going to happen after repeated dosing. And the idea came from knockout mouse that we may establish a new protocol for that. This is the data of a TCDD, that's the dioxin, and 3MC is the 3-methylchlorantolene. Both are AHR ligands, dioxin receptor ligands. And this 3D graph is CYP1A2. It has a baseline and then known as a AHR ligand inducible gene. And upper two is the wild mouse liver, and the lower two is AHR knockout mouse. So what happens is that in knockout mouse, the response is gone and the baseline is lower. But mainly, the key factor here is response is gone. So we thought what we did was that we challenged wild type and gene knockout mouse with the same protocol. Sorry, I had a. Okay, and so this is what we compare wild type mouse and knockout mouse. And we know that knockout mouse has a HR deficiency, so that there is a cascade of event lost by under the HR. So we thought this could be applied to a repeated dosing of a chemical idea. So instead of following up what's going to happen daily after repeated dosing in the liver, we thought we could give all animals for certain days, in this case 14 days. Again, 48 animals, we give all the same dose of a same chemical for 14 days and induce a chemically induced transgenic state, just like making a knockout mouse, not a knockout mouse, but something similar to what's going to happen by repeated dosing, so that we challenge the same protocol on the 15th day and compare with wild type without treatment and compare wild type with 14-day treatment and see what's going to happen in the response. And we first did with carbon tetrachloride. Again, the dose here is very low. If you are studying carbon tetrachloride and inducing liver lesions, you may give more, 10 times more or so. But in this case, we are looking at the signal, so we keep this absolute amount very low. But in any case, we selected 5 milligram per kilogram for 14 days to all animals and gave 0, 0.7, 2, and 7 on the 15th day and sampled 2 hours, 4 hours, 8 hours, and 24 hours after that, and compared with the animal who just got 0, 0.7, 2, and 7 without 14-day pretreatment. And in short, what happened was what we found that very few gene, but some gene showed enhanced response, like on the upper two panel. Left single versus repeated, which shows that the response after 15-day exposure was stronger than single exposure. And many genes lost the response after 14-day exposure. And we thought we saw that the baseline also changed. So we thought this is a epigenetic response induced by 14 days. So we simply went into monitoring what's going to happen by 14-day exposure in the DNA methylation of a whole genome by whole genome bisulfite sequencing and histone modification by chromatin immunoprecipitation sequencing. And well, methylation was simple, but in short, we had a positive control for this methylation monitoring. In short, we used JF1 Japanese type mouse and B6 European mouse. And the important thing is that there is a SNPs a lot in this JF1, so that in short, we can check which allele is from mother or father by looking at this DMR region, differentially methylated region, carefully, and we could develop a very reliable procedure to look into the methylation, and we applied to our sample. And for the immunoprecipitation for the histone, we couldn't do by ourselves, so we shipped out our sample to the contract lab. And of course, there are so many antibodies you can use, but we selected 2 pairs, one for activation marks and one for repression marks. And activation mark is the K4 trimethyl and K7, the 27 acetylation, and repression is 27 trimethylation and 9 remethylation. So we look at this control for the K4 and test chemical for K4 and control for 27 acetylation, and like pairwise. So we have a data made of 8 rows of data and see what's going to happen. And the data looks like this. So it is basically from the same sample and kind of same experiment, or you can match up with the 3D graph here. And this SRP9, what was that? Anyway, 9, which lost the response after 14-day exposure to the carbon tetrachloride. And if you look into the DNA methylation first, that's the lower half. The blue part is an unmethylated part, and this blue part is the CPG island of enhancer promoter region of this gene. And it looks like the treatment didn't affect the DNA status, methylation status, but if you look at the histone, it looks like the activation marks went down as it is shown by the red box here. And we went into clofibrate, again, 14 days of a clofibrate given to 48 animals, 70 milligram per kilo, and then challenged on the 15th day by 0, 10, 30, and 100, and followed 2, 4, 8, and 24 hours, and compared with untreated ones. And again, we looked at this PDK4, and we find out that the time course was the same, but the magnitude was lower after 14-day exposure. So we looked into the histone and DNA methylation. Again, the methylation didn't change. And this time, histone didn't change too. So we couldn't say anything from this data about this lower copy number. So we looked into the upstreams, and this is another software made by our collaborator in Systems Biology Institute, and we find that this PDK4 has AHR as an upstream, and then we looked at the AHR gene of this sample, and again methylation didn't change, but firstly, the 3D graph in the blue box shows that single exposure data shows some change after 2 hours and 8 hours, but you find that the baseline was 24 copies, but after 14 days repeated, it went to 10 copies. And if you look at the histone there, you find that the repression marks were increased. I mean, K9 3-methyl was increased by 14-day exposure. So we thought this could explain that AHR expression was lower because of this histone modification. And also we can find that PPAR is also upstream of this PDK4, so that we checked the PPAR levels of the same sample, and it was suppressed from 75 copy baseline to 45 copy baseline. And if you look at the PPAR gene histone here, if the red circle shows that the 27 trimethylation went up compared to control by chlorofibrate 14-day exposure. So we thought this could explain that PPAR alpha expression was suppressed, maybe by this histone modification. So we have to prove more carefully about the relation, but at least from the data here, we may be able to temporarily conclude that histone of a PDK for enhancer promoter region did not change by, so the PDK itself was not changed by 14-day exposure, but upstream genes, at least AHR and PPR alpha, was suppressed, possibly by this increase in the repression marks of the histone. And if you expand this idea to the cartoon like here, we can hypothesize from our data that chlorofibrate changed AHR histone and PPR histone and changed the level of AHR and PPR, so and changed the level of a PDK4. And so we are looking only at the messenger RNA and the histone change, so that we have not monitored the protein level yet, but we are assuming that this is a possibility here. Of course, we have so many question marks left here that chlorofibrate, we know that itself is a PPAR ligand. So how this changes the histone of AHR and PPAR alpha, we don't know. We have to think about that. Probably looking into the literature and many other data available. But in any case, we are trying step by step to draw out this type of connection between upstream factors and downstream factors. And eventually, we hope to connect to phenotype, prediction of the phenotype. And so I skipped this slide. And now I talk a little bit about next approach of our project, Perslon project, we name it. And that is an AI-assisted system for the comprehensive recognition of biologically significant alterations in one thing is this 3D surface graph. We are doing kinda automatically right now, but not perfect. We are developing deep learning type methodology as a collaboration with this System Biology Institute to select this surface change, because we have to select from 45,000 data set. But that we are starting right now, but we are also starting to look into histone patterns and DNA methylation patterns and related basic information. What I am trying to say is that this, again, we look into this graph of a histone, 8 lines of a blue colored, how do you say that? This is a tiny histogram, but graph, and lower 4 lane is DNA methylation. But unfortunately, currently, these findings are introduced by selected position and checking by human eye. And if you are familiar with this browsing system of this DNA, you know that this is a very, very narrow part of the whole genome. The whole genome is so wide that you cannot do by manpower to scroll or browse all the genes. So that's what we are trying to ask AI assisted system to do instead of us human eyes. And again, this idea starts all from the idea that this gene which shows the similar 3D graph pattern, like I show you in this box, there is a 6 gene here. You see it's very similar, and we think that this is a cluster like I showed you in this spherical globe with ripples. And currently, what the available method right now mainly would be to find this consensus sequence for the upstream factor is using the human mouse rat conserved sequences. But if you tried this method, in reality, there are so many genes that there are so many genes that conserved sequence cannot be found. You may encounter this this type of outcome so frequently. So we are now trying to develop a system that instead of using a human mouse rat to find the consensus sequence, we are trying to use this cluster based on the shape of the surface, so that in this case there are 6 genes, and align the CPG or the enhancer promoter sequence of this 6 gene and try to find where is common among these 6. And another idea is that, again, to look into the unmethylated region first, and then look at the histone pattern and identified which gene should be expressed in which organ and how the expression should be affected by the repeated exposure. So now we are looking for the change by our eyes, but we are trying to have an AI system doing for us. And one more issue, quickly go. This is the last slide, a few slides, that there is a region in DNA that liver gene and lung genes are aligned alternatively. This MAT1 is liver gene, and SFTPA is surfactant associated, which means lung gene, and MBL is again the liver gene, but the next again is FTA. This is the surfactant gene, and if you look at the data we have for the liver, of course, MAT1 is expressed like this, and MVL1 is also expressed, but surfactant is not expressed in liver like this. And if you look at the DNA and histone pattern, the surfactant gene of the liver, the area is red, which means that DNA is closed, methylated, and you don't find any histone to open up the area. Only the liver gene has it in the liver sample. And we have the lung data from the same animal. And again, in the lung, we don't have liver gene expressed. Instead, the lung shows surfactant positive here. And the same to the so the surfactant gene is expressed in lung, but not in liver, of course. But the data currently I have is only about the liver, histone, and DNA. So we are trying to have a lung data for histone and DNA, and we are hoping that the lung DNA has surfactant gene open and like that. So sorry about that. The conclusion for today's talk is that analysis of repeated dosing studies were facilitated by adding epigenetic data to the transcriptomic data. And genome-wide comprehensive analysis needs power up by the help of an AI system, that's for sure. And combination of a raw data-driven comprehensive analysis and information search, both supported by AI system, are effective for the predictive toxicology. So we are proceeding with this idea. And this slide is the acknowledgement for the development of the system, which started from year around 2003. And this is the current small group which supported the slides of today's talk. The people in the Toxicology Division of National Institute of Health Sciences and Systems Biology Institute, and the Laboratory of Bioinformatics, National Institute of Biomedical Innovation, Health and Nutrition. And the grant is mainly from National Ministry of Health, Labor and Welfare of Japan. Thank you for listening, and this is my talk today. Thank you.
Thank you very much, Professor Jin Kanong, in very informative and update research topic. And thanks again. And I have one question for you.
Yes.
We know that some of the DNA methylation in certain genes can turn on the gene, but sometimes the DNA methylation in the other gene probably can turn off the gene. I think the same situation could also occur in the histone modification. So in your experience, is there any possible the crosstalk or interact between the DNA methylation and histone modification? And which one is more dominant?
Oh, okay. Thank you for the question. In 14-day time range, unfortunately, we couldn't find DNA methylation change as a whole. We didn't look into a very tiny area yet, but as a gross level, we couldn't find DNA level yet. So that's why we had to stick to histone only for this research. But I think if we have enough grant to run, we may do 1 year or half year exposure, and of course we see DNA methylation. But more surprisingly, we thought that histone modification is rather quick and very strong in that sense. So that our idea is that histone modification starts first and then follows the DNA methylation for a more strong fixation of that change. But that part, we don't have the data. And we also don't have the data how fast the histone change goes back after stopping the exposure. And we are trying to have that data in the next 1 or 2 years. So we are doing very slowly, but the grant is not so big for us that we couldn't run so many studies at the same time. But thank you so much for your question. Yes.
Thank you. And one more question. So
Yes.
you focus on the liver organ, right? So after the repeated
Yes.
dose,
Well,
yeah. So
yes.
I just want to know, is there any possibility that the liver function be affected by the other organs?
Yes, yes, and vice versa. Today I had no time to express some data, but one good data we think we are now getting is brain is affected by liver, by cytokines, so to speak, and we thought we had a data that if you give the chemical through gavage, some chemical turns on gene in lung first.
Huh.
And if you make the same chemical inhaled by inhalation study, liver gene moves first, like opposite. So there's something that very funny thing going on, and of course it's not always applicable, but we think we found that such difference is there, and of course if you give a carcinogen, liver carcinogen, then the liver responds, but kidney doesn't, or vice versa. So the response is very different, but we are trying to have sequential signaling from one organ to the other. Yes.
Good.
Thank you.
Thank you. Is there any comment or question from the audience online?
Professor Kennel, may I have 2 questions?
Yes.
I'm Rachel Huang. Thank you for your excellent speech. I just have 2 concerns. The first question, can you hear me? Can you hear me?
Yes, very clear.
The first question is that we know that some toxicants or reagents, their toxicology are overlapping. That means that they may share the same targets of genes, proteins, and signaling pathways in response to environmental toxicants stresses. Which we may know according to the previous reports, or we don't know because we not detected. So my first question is that how to optimize the your experiments to make your funding more prices sensitive. Thus, I think it may be figure out it's those chemicals specific, unique targeting genes and pathways. That is the first question. So I would like to inform your supposed model that we can draw the each chemical has its unique ID in response to environment stresses in SARS. Just like human each person have our own unique ID. So another question is that in real world, the exposure of chemical you often not single but very complex, such as multiple chemicals may be intake into our body at the same time and can be metabolic in level to produce many of metabolites. And this can also influence the genes, targeted genes, and the proteins. So you proposed repeated this hypothesis model can be used to evaluate the multiple chemicals toxicity. So that's my two questions.
Okay, well,
A little bit long question,
yeah, right. But
but I'm so interested with your topic.
Thank you. So one thing is that I think one answer is that that's why we first looked into a very early response after a single exposure of a single chemical. And yet, even one chemical has multiple target anyway. So it's not simple. I just picked up a very easy to express very topic of one or two gene from one chemical, but normally it's like few thousand to some chemical really turns on 10,000. So one chemical has and a very early time course has a complex response. Yes, and one answer to you is that we could not look into metabolites. It's hard, and we don't look into kinetics either. It's very hard, but some chemical you see immediate response at 2 hours, which is normally a ligand to a receptor system. So TCDD, it immediately triggers some gene at 2 hours. But for example, pentachlorophenol or such very dirty toxicants, 2 hour, 4 hour, there is very few gene turned on. And suddenly after 8, a little bit, and 24 hours later, genes of NRF2 goes up suddenly. So we think between 2, 4, 8 hours, we think something is happening. It may be metabolizing the chemical to active compounds. But I'm sorry, we can only say that there's something going on, but and 24 hours it's high. But so we are looking at messenger RNA only, so that we have to think or guess from that data what's really happening at the beginning, and like metabolites and so on. So I think that's the next step we have to think about. But we think this is a beginning of your question answer. And the second thing is that we also touched a little bit of a combined exposure to chemicals. So we looked into single chemical A response, B response, and A plus B response. But and good thing is that we look into the gene response so that we can, and time course, so that we can look into the interaction of the two chemicals. But it's again very complicated. So we have some data, but not written the paper yet. I have to write it fast, but we only think about the combined exposure is understood by a very analytical way of looking into the molecular level response. But we need a little bit more data to really look into the details. we just started in that sense. Yes, thank you.
Thank you,
because,
Kenneth.
because the time is limited, I have to stop the discussion. Okay. So thanks again. Nice talk of the Professor Jin Kanong. And we have to take a break. You go get a drink and come back probably 5 minutes later. Okay. Thanks again. Thank you very much. We enjoy your talk.
Thank you.
Thank you.
Thank you.
So please come back at 3:11.
Okay, I see. Understand.
Okay,
After
shall-
your introduction, I will share my screen.
Okay. Dr. Ogra, are you ready?
Yes, of course.
Okay. So shall we move on now? The third topic will be biological and toxicological significance of tellurium. This session is chaired by Dr. Lin Yingqi, who is a rising star in our university. Professor Lin is currently director of Doctor Master Degree program in toxicology, Kaohsiung Medical University. She graduated from Taipei Medical University with a BS degree in pharmacy, and then a master's degree in clinical and administrative pharmacy from University of Iowa, USA, and her PhD from University of Minnesota in experimental and clinical pharmacology. She's also director of Substance and Behavior Addiction Research Center, Kaohsiung Medical University. So the podium is yours now, Dr. Lin.
Thank you. So it's my great honor to introduce Professor Yasumitsu Ogra today. Dr. Ogra obtained his master and PhD degrees in pharmaceutical science from Chiba University, Japan. He continued his postdoctoral training at National Institute of Industrial Health. He then worked in Chiba University as a research associate and associate professor, and in Showa Pharmaceutical University as a full professor. He then moved back to Chiba University as a full professor. He's now in toxicology and environmental health graduate school of pharmaceutical science at Chiba University. He is also a counselor of JSOT. His research interests include the elucidation and the toxicological effect of metals and metalloids, and the development of novel approaches for speciation of biotrace elements and their metabolites. He will introduce us the biological and the toxicological significance of tellurium and an industrial important metalloids today. Please join me in welcoming Dr. Ogra.
Thank you very much, Dr. Lin, for your kind introduction. So may I share my screen? At present, not allow me. Please allow me to share my screen. I don't know why. Okay, thank you. Is this okay?
Looks
Yes,
good.
Can you hear me? Can you hear me? Okay. Okay, so let's have my presentation. First of all, I really thank Professor Lee and other TSTA members for giving me opportunity to talk about my recent work in such an interesting webinar toward to the ASIATOX 10. Today's my talk is about tellurium. So this is today's agenda of my talk. I think tellurium is not so familiar to almost of you. So first, I briefly introduce the element of tellurium. And then I'll talk about identification of the tellurium metabolites in the plants, and also identification of the urinary tellurium metabolite in animals. The tellurium is a member of the group 16 elements consisting of oxygen, sulfur, selenium, tellurium, and polonium. And tellurium is a non-essential element for the animals and the plants. As you already know, oxygen and sulfur is essential elements for the animals and the plants. In the case of selenium, selenium is only essential for the animals, not essential for the plants. But in the tellurium, maybe also polonium is also non-essential elements for animals and plants. And tellurium is called metalloids or semi-metal, which has a unique physical and chemical properties. Metalloid or semi-metal means element which has physical chemical properties, both transition metal and typical element. So this unique physical and chemical properties, the tellurium is widely used in the electronics and chemical industries. Also, due to this unique physical-chemical properties, tellurium has also unique biological and toxicological effect. So, in particular, some types of DVD contain the tellurium. I'll show you the actual content in the next slide. In the baby, he likes a greeting item. This baby is my daughter. At that time, my wife said to me, she's okay or not, she's safe. At that time, I have not enough answer to convince my wife. So this is one of my motivation to elucidate tellurium toxicity. And in Japan, used DVD also often used in the home garden as a scarecrow. I'm not sure that this unique application of DVD is popular in other Asian countries. Anyway, tellurium in the DVD would be dissolved with rainwater and then incorporated into tomato, eggplants, or cucumber in a home garden. So I think tellurium could be a risk to human health via this kind of the vegetable in the home garden. This slide shows the actual concentration of the metalloid in the optical discs. There are two types of optical discs. One is a phase change type, which made from the inorganic materials. The other one is a non-phase change type. This is made from the organic materials. In the case of the phase change type of DVD, actually germanium, antimony, tellurium, and bismuth is detected in its disk. As you can see, actually tellurium is a substantial amount of the tellurium is containing in the phase change type DVD, such as DVD RW and DVD RAM. We also paid much attention to the tellurium in the accident of Fukushima Daiichi nuclear power station in March 2011. You may know the nuclear fission of uranium generates this kind of the radionuclides, such as xenon and iodine and cesium. In addition to these radionuclide, radioactive tellurium is also released to the environment. By the nuclear fission of the uranium, this around 130 of molecular mass, such as xenon, iodine, is generated. But in the case of tellurium, tellurium also has around 130 of molecular mass. So these radioactive nuclide of tellurium is also released into the environment. So roughly estimated that the amount of the released radioactive tellurium are more than 10 to the power of 17 BQ. So as I mentioned, tellurium must be one of the environmental risks. However, at present, no toxicological and toxicological, even biological information of tellurium are limited. So we intend to obtain the toxicological and biological effect of tellurium in terms of its metabolism in animals and plants. So when we expose the inorganic tellurium, this is a chemical form mainly found in the nature. In the inorganic salt exposed to the animals and plants, so tellurium is a metalloid, so the tellurium could be form organometallic compounds in its metabolism. So we identify the tellurium metabolite. That means we can know the metabolic pathway of tellurium. So we identification of the tellurium metabolite in animals and plants are needed to fully understand the tellurium toxicity. The first, I'd like to talk about the identification of the tellurium metabolites in a plant. Selenium is the same group element of tellurium, and the selenium is non-essential element. But some plants are known as a selenium accumulator. Selenium accumulator means sulfur-rich plants that is a large metabolic capacity of selenium and sulfur. I think maybe tellurium is also metabolized in this selenium accumulator plants because selenium accumulator plants may share the metabolic pathway, not only selenium but also the tellurium. So tellurium could be accumulated in the selenium accumulator plants. These plants are used in our studies. The upper plants are selenium accumulator plants, such as garlic and Indian mustard, broccoli, and nozawa. Nozawa is the same Japanese original, maybe a Japanese original species, and which is the same family plants of Indian mustard and plants. And rucola is also known as a selenium accumulator. Lower plants is a non-selenium accumulator plants, but these are also frequently used in our food. So we use the garlic because garlic seems to be a suitable plant for the identification of the tellurium metabolite due to the following two reasons shown in the next slide. Broccoli and garlic actually accumulate. This is a simple experiment to expose the selenium and tellurium in these plants. Broccoli and garlic actually accumulated as selenium. But maize is a typical selenium non-accumulator plants. This is not accumulated. This are not accumulated the selenium. Contrary to the selenium, tellurium was not accumulated in the broccoli. Broccoli is although broccoli is known as a selenium accumulator. But in the case of the garlic, garlic is actually accumulated to tellurium. So we think garlic is most suitable plants to evaluate the tellurium metabolites in the plant. In addition, garlic is very easily cultivated under the hydroponically conditions shown in the next slide. This is an experimental design of our tellurium project. The garlic bulb was planted on the PET bottle cultivation flask. And this is easily hydroponically cultivated and easily expose the unknown amount of the tellurium. In this case, we used 50 to 100 to 100 micromolar of the tellurium as a tellurate. And 2 weeks for the exposure, we harvested the garlic and divided into the three parts, leaves and bulb and roots, and analyze tellurium concentration and identify the tellurium metabolites. This slide shows the analytical technique we used. For the screening tellurium metabolites, we used a speciation analysis consisting of the HPLC and inductively coupled plasma mass spectrometry. ICP-MS. ICP-MS is a very famous elemental mass spectrometer And this spectrometer has a high sensitivity and under the complex matrices means very robustness against complex matrices. And in this case, we use HPLC as a separation technique, and ICP-MS is used for the detection technique. So this hyphenated technique called LC-ICP-MS. However, ICP-MS is not applicable to identify the unknown metabolite. Exactly say if we can available the authentic standard compounds. In that case, we can match the retention time unknown compound with authentic standards. But we have appropriate authentic standards to identify the unknown compounds. In that case, ICP-MS is not effective to identify the unknown compounds. In that case, we must use the LC-ESI tandem mass spectrometry to identify the unknown compounds. This is a result, the first result obtained by the LC-ICP-MS. 3 pteridine metabolites were detected. The tellurate is an exposed chemical form. In addition to the tellurate, that means the intact form, we detected 3 unknown pteridine metabolites. Unknown means there are no authentic standards matching with these detention times. So in the next step, we try to identify them by the electrospray mass spectrometry. Among these 3 compounds, unknown 1, 2, and 3. Unknown 3 was the most easily isolated, so this was the first target of us. Because in the case of the analysis by the electrospray tandem mass spec, in that case, we must partially purify the compounds because ESI mass spectrometry is less robustness than the ICP-MS. So unknown compound 3 are most easily purified comparing to the other metabolite. So we choose this compound as our first target. This show the theoretical tellurium isotopic pattern. Tellurium consists of 8 stable isotopes in nature from 120 and 130. Hence, a molecule containing a tellurium must show that this isotopic pattern. At first, we detected the tellurium species at M/Z 250. This is a result of a mass spectrum of unknown compound 3. To obtain the further information about the molecular structure of this unknown compound 3, the most 3 abundant peaks, that is 250 corresponding to the tellurium 130, and 248 corresponding to the 122, and third one, these 3 peaks are subjected to the MSMS analysis. So due to the time limitation, I cannot say the detailed assignment of these fragments, but we are able to identify this compound as a tellurium metabolite of unknown compound 3. This structure is methylphthalocystine oxide. Methylphthalocystine means the major structure of this compound is cysteine. Now, in the case of the cysteine, this part is sulfur. Phthalocystine means substitution, tellurium derivative substituted sulfur to the tellurium. And then this tellurium is methylated and also oxidized. So this compound is named methylphthalocystine oxide. To confirm the structure of the tellurium metabolite, we chemically synthesized this compound. Actually, we synthesized methylphthalocystine. But this compound was easily oxidized under the ambient condition. According to this strategy, we can get firstly we can get this compounds methyl cysteine, but this compounds are easily oxidized to this form. And this compounds actually gave us the same mass spectrum as the unknown compound 3. So we think our compounds could be unknown compounds could be this compound. But to confirm the structure, we check the chromatographic behavior. We partially purified unknown compound 3 and spiked with a chemically synthesized compound. The spike which chemically synthesized methylcysteine oxide specifically increased the peak corresponding to unknown 3. This result solidly showed the unknown compound 3 is methylcysteine oxide. However, as I already said, I think maybe methylcysteine oxide is an artifact to produce the ambient condition. So real plant tellurium metabolite could be a methylcysteine, not oxide form. We have identified unknown compound 3 as a methylcysteine oxide in the elution profile. So next target is unknown 2, these compounds. In this case, we use the LC-ESI high resolution mass spectrometry. I show only MSMS spectrum. These fragments can be assignable to these compounds. This is a cysteine S-methylterulosulfide. I think this is also artifact compounds found in the plant samples, because this part is cleaved from the methylterulocysteine, and this cleaved methylated tellurium is sequestered by the cysteine. But we can detect it. We can identify this compound as an unknown too. At present, we have identified two of three tellurium metabolite. One is this, the other one is this. The last one is the unknown one. However, at present, we have not reached the structure of this unknown compound one yet. So we also check the tellurium metabolite in other plant species, including selenium non-accumulator plants. At first, we said we tested at least 8 or 9 plants, but since here I only show the garlic result. So in the next slide, I will show the result of other plants. This is a speciation of tellurium in the leaf extract of other plants obtained by LC-ICP-MS. But in garlic, 3 tellurium metabolites are detected. However, all other plants we tested can metabolize tellurate to only one metabolite, unknown 1. But unfortunately, unknown 1 is not identified yet. But thus, I think unknown 1 is a common tellurium metabolite. As you can see, this unknown 1 is detected broccoli, Indian mustard. Upper plants is a selenium accumulator, and lower plants are selenium non-accumulators. But we can detect it unknown 1 compounds, both selenium accumulator and non-accumulator. So we think this unknown 1 is a common tellurium metabolite in the plants, and maybe important metabolite of tellurium in plant. Now we continuously tackling to identify this compound, but at present we have not reached to this structure. So I'd like to summarize the first part of my talk. We continuously try to identify the unknown one, but at present, garlic can metabolize inorganic tellurium into 3 metabolites. Two of them are identified as indicated below. One is a methylcysteine, the other one is a cysteine S-methylptyrosulfide. So methylcysteine is firstly identified in higher plants. I think this is a very important result because this is the first case to show the tellurium can be assimilated in the plants to form the tellurium carbon organic bond in its in the plant. And other plants metabolize tellurium into the unknown one. So we think unknown one is a key metabolite of tellurium in plants. So we continuously try to identify this compound. So the second topic is identification of the urinary tellurium metabolite in rats. In these experiments, male oyster rats have freely access to the drinking water containing the inorganic tellurium for consecutive 8 days. The urine was collected and the urine sample was analyzed by the LC-ICP-MS and LC-ESI-MS to identify the animal tellurium metabolites. In this elution profile, tellurium elution profile, as you can see, only one tellurium metabolite was detected by the LC-ICP-MS. We tentatively named this compound urinary tellurium metabolite. But in the case of the selenium, we can detect it two urinary selenium metabolite. In this case, the animal can take a selenium beyond the nutritional range. Within the nutritional range means within the adequate nutritional level. The selenium sugar is a major urinary metabolite of the selenium. The selenium sugar is a very unique compound. Selenium is attached with a sugar moiety. But in the sugar, this sugar part is a specific N-acetyl galactosamine form. This is a major urinary selenium metabolite. The other one is a TMSE, trimethylselenium ion. This compound is appear when selenium is ingested beyond the nutritional level. So we think this is simply methylated to reduce the tellurium toxicity. As you already know, selenium is essential for the animals, but very toxic element. Within the nutritional range, selenium is mainly metabolized to the selenium sugar. But beyond the nutritional range, selenium is metabolized to the simply methylated form as trimethylselenium ion. So we think the retention time of this tellurium compound is very close to the TMSE. So we think these compounds, we speculated these compounds could be a trimethylselenium ion. So we chemically synthesized trimethylselenium ion. As I already suggested, tellurium has 8 naturally occurring isotopes, and we can detect it, this isotope pattern at M over Z around 175. 175 means a molecular mass of the trimethylselenium ion. So we can successfully synthesize these compounds, and we use this authentic standard compounds to check the chromatographic behavior of unknown tellurium compounds. We spiked chemically synthesized TMTE into the urine samples containing the unknown tellurium metabolite. As you can see, the spike specifically increased the peak of unknown tellurium metabolite on the multimode gel filtration column. So we used another type of HPLC column to solidly identify unknown tellurium compounds. In this case, we use the cation exchange column to check the chromatographic behavior of urinary unknown tellurium metabolite. As you can see, as expected, the spike specifically increased these unknown peaks. That means, according to that, these chromatographic behaviors on two different separation mode. So this urinary metabolite could be trimethyl telluronium ion. So this is a conclusion of second part of my talk. It is reported that selenium is metabolized to urinary selenium metabolites such as the seleno sugar and the TMSE. In this case, the methyltransferases responsible to produce TMSE are already identified our group. That enzymes are TPMT and INMT. TPMT is a thiopurine S-methyltransferase. INMT is indole ethylamine N-methyltransferases. These two methyltransferases are needed to form the tolerably methylated urinary selenium metabolite. So TPMT is responsible to first and second methylation, and INMT is responsible to the second and third methylation. Anyway, to form the polymethylated form, these two enzymes is needed. We speculated TMT is also these two methylated compound methyltransferases. But at present, we have not so solid experimental result. These two methyltransferases actually needed to form the polymethylated form of tellurium compound. But as I already shown, no tellurous sugar was excreted into the urine. Only selenium is metabolized to the seleno sugar. No tellurous sugar is identified in the urine samples. At present, what kind of the enzymes responsible to form the seleno sugar? But this unknown seleno sugar synthetase can discriminate tellurium from the selenium. However, these two enzymes could not distinguish tellurium from the selenium. That means that within the nutritional range, selenium is an essential element. So essential element must metabolize the specific metabolic enzymes. But these enzymes strictly discriminated essential metal, that means the selenium, and non-essential element, tellurium. But beyond the nutritional range, at that time, the selenium recognized as a non-essential element because selenium is beyond the nutritional range. In that case, these two enzymes methylate selenium without discrimination of the non-essential element of the tellurium. So I think maybe at the enzyme levels, the essential element and the non-essential element could be discriminated. Even in the selenium, within the nutritional range, selenium is metabolized by the specific enzyme. But beyond the nutritional range, selenium is metabolized as the same enzyme as a non-essential element. So let me conclude my talk. The first point is we identified a novel tellurium metabolite in a garlic and rats. Garlic can assimilate tellurium to methyltellurcysteine. And rats excreted tellurium into the urine as a trimethyl telluronium ion. And the major tellurium metabolite in rats has not been identified. Anyway, these unique tellurium compound as identified as a pteridinium metabolite in garlic and rats. As further work, we should identify the unknown one in plants. But considering the eco-circulation of the pteridinium, in this experiment, we only evaluate how inorganic pteridinium is a metabolite in animals and plants. But the eco-circulation of the pteridinium, plants actually synthesize this unique pteridinium-containing amino acid. And animals excreted this unique pteridinium metabolite. And this urinary metabolite could be exposed to the plants, and this unique pteridinium-containing amino acid also exposed to the animals by food and vegetables. So we think in the further experiment, we must evaluate the toxicological effect of methylphthalocystine and trimethyltelluronium ion, and also unknown one. I'd like to thank my staff and the student for their contribution to our pteridinium project. This university, my ex-affiliation, and now current affiliation at the Chiba University. So we thank many staff and the student for their contribution to this work. And also I thank for these founding agencies for their financial support. And then thank all of you for your kind attention. Thank you very much.
Thank you, Dr. Ogra, for the wonderful talk. So I think we have few minutes for questions. Anyone from the audience want to ask question?
Excuse me.
Yes, please.
May I have two questions?
Thank you.
Okay. The first, thank you for your wonderful speech. I just have the first question is that I'm a little bit curious. What is the relationship of garlic experiment and rat experiment? And the second question is that I'm not sure why you choose garlic as your target research, because garlic has its own detoxification ability. So I'm still have this concern why you choose this garlic. May I understand that the garlic, that means more garlic may have the more ability to avoid the TE stress from our body? Thank you.
Thank you very much for nice question. The first question to my answer is rat and garlic is completely independent. We use the garlic and rat as a model plants and model animal to identify the plants pteridinium metabolite and animal pteridinium metabolites. So there are no reasonable relationship between the garlic and the rats. This is a completely independent, only for the identification of the pteridinium metabolite in animals and in plants. We use these two as a model plants and model animals. That's my answer to the second question is in this experiment, garlic is a very convenient model plant because we can easily hydroponically cultivate it and easily control the exposure concentration. And we use the normal garlic samples. Garlic, it's not a specific species. It's a normal garlic, but if we can get if we purchase the garlic in market, the garlic is not germinated by the plant hormone treatment. So we purchase a specific garlic can germinate for experiment. But I think that we use the normal type of the garlic, not a special species. Is that okay for your question?
Yes, thank you.
Yeah, I think we have an audience who raised his hand.
Hi. Hi, Professor.
Hi,
Yeah.
Professor Chen.
Yeah, this is Chen Ying. Yeah. Very nice talk.
Thank you very much.
Yeah, thank you. I would like to ask one question about the how to identify the new species of Pinanian in the animal, because you try to use ICP-MS to identify the new species. I think it's quite it's quite difficult. It's a big challenge to identify the new one. So how to how could you do that?
In this case, we can also identify these compounds by electrospray IMSMS. But in this case, fortunately, we can easily get the authentic chemically synthesized TMTE that used as an authentic standard. In that case, we check the retention time of unknown compounds. To ambiguous identification, we use two types of HPLC column. Both columns show the same retention time between the unknown compounds and authentic standard. In that case, I think maybe acceptable to identify the ICP-MS. But it is in the case of the difficult to get the authentic standard. In that case, we must use the electrospray tandem mass spectrometry to solidly identify the unknown structures. But this is I think maybe this is a lucky case to we can use the ICP-MS.
Yeah, thank you.
Thank you very much. See you next week, Professor Chen.
Yeah.
So I have a follow-up question
Thank you very much.
Regarding the Dr. Chen's question. So have you used the analytical method in more complicated samples? For example, like tissue samples, will it still work?
Yes, but in the garlic sample is not so complicated because the matrix is very, very simple. But you suggested that tissue samples such as liver and brain, and the urine sample is also consists of the very, very complicated matrices. In that case, you must purify the sample to detect the electrospray tandem mass spectrometry. But in the case of the ICP-MS can acceptable very, very complicated matrix samples with a high sensitivity. So we can use the ICP-MS as a screening technique. But for the solid identification, we must use electrospray tandem mass spectrometry. But in that case, we must purify the samples comparing to the ICP-MS samples. So we can, if we can clearly purify the samples, we can use the electrospray mass spectrometry to identify the unknown compounds. For example, urinary selenosugar. Selenosugar is also our work to identification of the selenosugar was our work. In that case, we use the urine samples. So we use the huge amount of the urine sample to identify the purified selenosugar. It's tough work, but we can get the we can use the electrospray if we can purify the samples to qualify the electrospray acceptable range. It's okay.
Thank you. Okay. Do we still have more questions from the audience? If not, then I think I'll just pass the floor to the next speaker. Thank you, Dr. Ogura.
Thank you very much.
Thank you, Dr. Ogren and Dr. Lin. Let's move on to the fourth session, which is chaired by Dr. Jun Kano. I think everybody knows Dr. Kano now because he's been introduced early on. So if Dr. Kano is ready, could you introduce the speaker and to chair the full session? Thank you.
Yes, thank you. The fourth topic is the possible roles of autophagy in nanomaterial toxicity and safety assessment related alternative testing strategies. Take the silver zinc oxide nanoparticles, for example. And could you next slide, please? Yes. The speaker is Dr. I'm sorry, I'm not good at reading this. Yin Jian Wang Sensei. I think I'd like to introduce you again. Distinguished Professor of the Department of Environment and Occupational Health, College of Medicine, National Cheng-Kung University, and his education is molecular biology from the National Taiwan University, and currently, this is the important part, President of Toxicological Society of Taiwan, and Dr. Wang's research interest is molecular toxicology and nanotoxicology. Dr. Wang, please, the floor is yours for your presentation, please.
Okay, thank you very much. I will share my slide. So I'm not allowed to share my slide. Please, the control screen. Oh, okay. Can you see me? See the, is that okay?
I don't see your slide yet.
No, not yet.
No, we
Not
can't.
yet.
Yeah.
Is that okay?
Yes. Thank
Okay,
you.
good.
Yes. Perfect.
So good afternoon, everyone. Thank you very much for giving me this opportunity to share with you some of our recent work regarding the autophagy, which we believe that can be an early biomarker of neurotoxicology, both in safety assessment and also in the alternative testing strategy. And I will take the silver and zinc oxide nanoparticle, for example, from the point of view of neurotoxicology, which is a very hard research topic during the past 2 decades. So as we know, the definition of the nanomaterial, the nanoscale material, or you can also call it the nanomaterial, nanoparticle, or engineered nanomaterial, engineered nanoparticle, was defined as the dimension between 1 and 100 nanometer. And the novel opportunity can provide novel application. So recently, the nanotechnology has rapidly promoted the development of a new generation of the industry or commercial products, such as the energy, such as the nano devices, cosmetic, medicine, and so on. However, the application of nanotechnology also raised some concern of the human health and also the human safety. And until now, the impact of nanoparticle on biological systems, especially the toxic effect, are still limited. So currently, several important nanomaterials has been applied in the biomedical fields for the purpose of diagnosis and therapy. For example, the carbon-based nanoparticle and silica-based nanoparticle for drug delivery, and the metal and metal oxide-based nanoparticle for antibacterial and also for imaging and therapy. And some of the liposomes and the polymer-based nanoparticle is already be used in the clinic. So basically, once we were exposed to nanoparticles, either from inhalation, skin contact, or injection, the nanoparticle can translocate into other organs, either through the circulating system or through the lymphatic system, and even penetrate the BBB and into the NSA nervous systems. That means that nanoparticle can distribute all over the human body, lead to systemic toxic effects. Because the epidemiology and the human volunteer study is limited, so the hazard assessment of nanoparticle can only rely on some of the toxicity tests to predict their health outcomes. So thanks for the contribution from nanotoxicologists during the past 2 decades. So we now understand that the physical and chemical properties of nanoparticle can strongly affect their cytotoxicity. In general, the small size is more toxic than the large size, and the positive charge is more toxic than a negative charge. And the surface coating is kind of case by case, and the composition that the rapid release of metal ion is more toxic than a slow release one. And the morphology with higher aspect ratios is more toxic than the other shape. So according to the REACH regulation, the safety assessment of nanoparticle depends both on the exposure level and also the toxic level. So as a toxicologist, our duty is trying to determine the toxic level of some nanomaterials. So if we consider the nanotoxicology and the nanosafety during the manufacture and further biomedical application, the idea that safety by design probably can help us to exclude some of the potential toxic materials in the very beginning and ensure the safety of the the final nano products. So the traditional safety assessment of chemical based on animal study. However, the EU, the European Union, promote many years for the development of the alternative testing. The so-called 3RS stand for replacement, reduction, and refinement. One of the reasons is because the traditional risk assessment of adverse outcome pathway in the selection of endpoint. So understanding the so-called toxicity pathway, understanding the mode of action and the whole process of adverse outcome pathway is a fundamental to the alternative animal testing. So before we can develop a new prediction model for safety assessment, I think the first step we need to do is to understand as much as possible the underlying toxic mechanisms of nanoparticles. So you can see the oxidative stress information and the release of metal ions, activation of the inflammasome, and also the lysosome, the injury. I think it's play fundamental role in some of the nanomaterials induced toxicity. So in our recent published review article, we try to summarize some of the mechanisms of nanomaterial-induced autophagy and subsequent systemic potential toxic effect. So you can see oxidative stress induced by nanoparticle can trigger the ER stress and cell death by apoptosis. And it can also induce the mitochondrial damage and trigger the inflammation in chromosome activation. And the damage in mitochondria can also activate the autophagy. If the nanoparticle was taken up through the endocytosis, it can also lead to the lysosome dysfunction and the autophagy dysfunction. And the cell probably can die eventually by another kind of cell death type called the autophagy cell death. So I will divide my talk into three parts. The first one is regarding the lower autophagy in silver-induced toxicity. And the second one, I'm trying to find out the autophagy-related pathway in zinc oxide and UVB-induced skin damage. And the last one, I asked one question: could autophagy be an early biomarker of nanomaterial-induced toxicity? So all this in vitro, in silico, and in vivo study aim to address the narrow safety assessment with a faster, cheaper, and sensitive ways. So regarding the autophagy, you may know that the Nobel Prize for Physiology and Medicine in 2016 went to Japanese scientist Yoshinori. Actually, autophagy can capture the intracellular component such as the damaged protein and damaged mitochondria into autophagosome, and then bring them to lysosome, and where they were degraded and recycled. So in general, autophagy can be induced by a variety of stressors such as the starvation, DNA damage, protein aggregation, mitochondrial damage, and ER stress. And the purpose is to prevent the cell death through the generation of ATP energy, through inhibition of the p53 signaling pathway, or through inhibition of apoptosis. But in some of the situations, such as the continuous stress, such as the defect of the p53 and apoptosis pathway, the autophagy can also lead to so-called the autophagic cell death. And we do have some experience about autophagy. So in 2007, about 10 years before the Nobel laureate Yoshinori, we published our first study regarding the environmental toxicant, the arsenic trioxide-induced autophagic cell death in glial cell. And since from then, we focus our study in autophagy, not only in environmental toxicology, also extended to cancer research and neurotoxicology. And we do publish a lot of scientific papers in several important journals. But today I'm going to focus only my talk only in neurotoxicology. So basically, autophagy can be induced by oxidative stress and inflammation. And the induced autophagy can also feedback inhibit the inflammation and oxidative stress. And the idea that nanomaterial-induced toxicity can, through the regulation of autophagy, has attracted a lot of attention recently. And however, the underlying mechanisms and also the biology response is still unclear. So more recently, there's a review article entitled "The Autophagy Modulated by Inorganic Nanomaterials," and you also summarize some of the important nanomaterials such as the iron oxide, silver, gold, carbon-based, and silica-based nanoparticle. When I look at more detail into the circle right here, and I think I see some of the the signal cascade triggered by silver, and almost all the biological response have been reported by our previous study. And I think it's a in 2014, we published our first study regarding autophagy in cell culture model. And I think our lab is, I think, the first one that proved both apoptosis and autophagy effects are involved in the cytotoxic effect in fibroblasts triggered by silver. And I think it's a very important finding because until now, this paper has been cited for more than 150 times. And why is the safety of silver so important? Because silver nanoparticle is the most common commercialized nanoproducts on the market. It can be applied in textile, the food industry, medical devices, cosmetic, and so on. So increase of human exposure lead to increasing the public concern. So you may know silver has an excellent antimicrobial property, and the release of silver ion from the nanoparticle accounts for much of the bacterial killing effects. But meanwhile, it might also trigger the cytotoxicity. And actually, the bacteria do not take up nanoparticle, but the mammalian cell can take up the nanoparticle and fusion them with the lysosome, where the signal ion, the silver ion, can be quickly released inside the lysosome through the mechanisms we call it the Trojan horse effects. So in our previous study, we demonstrated the serial uptake of silver nanoparticle by using the TEM flow cytometry. So you can see the internalized nanoparticle right located in the so-called autophagosome. You can also see the complexity of the cell can be induced in exposed to silver by using the flow cytometry. So we found the silver can induce morphological and biochemical markers of autophagy in fibroblasts, evidenced by the AO staining, also the immunofluorescence staining, and the protein marker of autophagy such as the P62, LC32. So all these results confirm the induction of autophagy. And at that time, induction of autophagy in cell triggered by silver has never been reported before. So it encourages us to further find out the underlying mechanisms. So in this regard, we found the treatment of silver can also induce the intracellular ROS generation and decrease the reduced form of glutathione in the cell. And silver can also induce the ER stress by using the ER trigger diet. So the most interesting finding in our study, in this study, is that treatment of silver can induce autophagosome formation and the lysosome dilation by using the double immunostaining of the by using the anti-LC3 antibody and also the anti-LMP1 antibody and the lysosensor. So you can see here the separation of the autophagosome and the dilated lysosome indicated the dysfunction of autophagy. So we summary our previous study briefly in this figure. You can see that silver can be taken up and trigger the oxidative stress. So the ROS could be an initiated event that can trigger ER stress and subsequently autophagy. And silver can also trigger the autophagy accumulation and the impair of lysosome. So both contribute to the autophagy dysfunction. And in 2016, we published a review article in Neurotoxicology entitled Mechanisms of Silver-Induced Toxicity and Important Role of Autophagy. So in this table, we put our finding that defective autophagy, in addition to several well-known toxic mechanism of silver, that defective autophagy could also play an important cellular and molecular mechanisms underlying silver nanoparticles toxicity. And recently, we extended our study from in vitro to in vivo by using an in silico model to determine the mechanistic study of silver-induced toxicity. So you can see in this flow chart, the M1 of our study is trying to apply the so-called KDD process and the knowledge discovery in database to construct a decision tree model and for ranking the strengths of the dose, cell type, and silver type, and duration. So these preliminary results can help us to achieve the M2, the mechanistic study, and also the M3, the in vivo study. And in the field of neurotoxicology, we have an agreement that before we conduct any toxicity assay, we have to provide sufficient or essential some of the physical chemical properties of materials because the physical chemical property such as particle size, size distribution, charge, surface coating can strongly affect their toxicity. So in this study, we apply 4 different kinds of silver, the small size and large size of citrate coating, and also the small size and large size of amine coating, characterized by the TEM, EDX, DLS, and UV visible spectrophotometer. So we conduct the cell viability assay of several different cell lines derived from different organs, such as the lung, liver, skin, kidney, immune cell, and also the intestine cells. So those results of each cell type treated with 4 silver types at the indicated time and the indicated dose. And this figure shows the decision tree model built for predictive ranking of the attributes assessment relevant to the assessment of silver-induced cytotoxicity. So you can see the exposure dose is most contributor is the most important attribute when compared to the other attributes, followed by cell type, silver type, and the exposure time. So you can also reconstruct the decision tree model, if you want to look into more detail of certain attributes. So for example, the silver nanoparticle types can be divided into particle size and the surface coating. So the overall results told us that the order of the 4 toxicity relevant attributes as the following: the exposure dose is most important, followed by cell type, silver type, and exposure time. And the order of the cell susceptibility toward the silver-induced cytotoxicity is as the following: the intestine cell is most sensitive, followed by lung cell, followed by kidney cell, liver cell, skin cell, and immune cell. And the toxic potential of the 4 silver particle type, I think it's more complicated. It depends on exposure dose and also the cell type. So regarding the mechanism study, we assume that both the apoptosis and autophagy can be activated by the high dose exposure of silver, and the cell cycle arrest and cell senescence can be activated under the low dose exposure of the silver. So to test our assumption, we analyze some of the important protein exposure involved in the regulation of cell cycle arrest, apoptosis, and autophagy. So you can see in this figure, we found the silver under a non-cytotoxic dose, the low dose of the silver exposure need to hasten G2M cell cycle arrest. And if we prolong the exposure to silver, also under the low dose exposure, we found that it can lead to the senescence and the inhibition of the cell proliferation detected by the senescence-associated beta-Gale staining and by the chronogenic assays. And several important proteins involved in the regulation of cell cycle arrest and regulation of the apoptosis and autophagy also kind of support our assumption that low dose exposure of the silver trigger more the cell cycle arrest, and high dose exposure of the silver trigger more the apoptosis and autophagy effects. So in the in vivo study, we apply 2 small sizes of silver with the different surface coating, the citrate and amine coating, and determine comparing their acute toxicity through the IP injection in mice. But to our surprise, the acute toxicity of the citrate coating, the negative charge, is much, much higher than the amine coating, the positive charge, which is against the general rule of the neurotoxicology. Because I mentioned it in the very beginning, in general, the positive charge is more toxic than the negative charge, but the detailed mechanism is still unclear. And to monitor the organ distribution and accumulation, we apply the fluorescence dye that can, conjugate on the surface of the silver and detected by IVIS. And we found among the 5 organs named, the most distributed organ is the liver, followed by kidney and lung. And if we extend the exposure time to 2 days, 7 days, or 14 days, and we found the most accumulated organ is the spleen, followed by liver and kidney. So this table shows some the biochemical data from the control and from the silver treated mice. So you can see a dramatic increase of the ALT and AST, indicating a severe liver damage. The kidney function, I think, is still fine, but we also find a dramatic increase of the amyloid acid activity, and along with the increased level of serum glucose, so indicating a severe pancreas damage. And in this figure, we found the organ morphology of the kidney, liver, and spleen didn't change a lot, but the morphology of pancreas do change a lot. So the silver exposure can lead to the collapse of the pancreatic structure and severe damage. And the whitened pancreas represent the acute hemorrhagic pancreatitis. So we summarize some of the important points here. First of all, the cytotoxicity, oxidative stress, apoptosis, and autophagy, lysosome dysfunction, all contribute to the cytotoxicity of silver in fibroblasts. And in the in silico decision tree-based catalytic process could be applied to identify the attributes related to silver-induced toxicity. And the silver could exert toxicity in so-called dose cell organ type, as well as the particle type dependent manner, both in vitro and in vivo. And I think the most important finding in this study is the IP injection of lethal dose of silver can lead to severe damage to pancreas. However, the detailed mechanism is still unclear. And the second part of my talk is autophagy-related pathway in zinc oxide and UVB induced skin damage. And actually, zinc oxide has been widely used in cosmetics, especially in the sunscreen, for the protection of the UV light. However, the safety issue of zinc oxide is still a concern, especially under the circumstance that the intake of skin barrier was damaged by the UV light. So we assume the skin damage induced by zinc oxide and UVB could be related to the autophagy autophagy pathway, based on the recent finding that both metal and metal oxide nanoparticle can trigger autophagy. And in our recent study, we demonstrated the skin damage induced by zinc oxide combined with UVB can be mediated by pyroptosis through the inflammasome, autophagy, and exosome pathway. I think the most unique contribution of this study is that we provide a novel, the so-called key event relationship during the adverse outcome pathway. So I've mentioned it in the very beginning that understanding the process of AOP is the fundamental for alternative animal testing. So the chemical or the nanoparticle, the nanotube can all serve as the chemical initiator that bind to the molecular initiating event, trigger key event and key event relationship, and lead to adverse outcome, such as the organ dysfunction, more formation, disordinity, and toxicity. So in our case, the input is zinc oxide and UVB. The output is skin damage. So in a traditional standard animal study, we don't know any detail inside the body. But if we can extend the traditional animal study and understand the process of AOP, we have a chance to develop new alternative methods and making the black box into gray box or even the open box. And the final goal is the regulatory acceptance. However, until now, there's still a long road to go to animal-free testing. And the inflammasome is recently been recognized as a key player in nanoparticle-induced inflammation and pyroptosis. So you can see the nanoparticle can activate the inflammasome, leading to the formation of the transmembrane protein of GSD-MD and secretion of the IL-1 beta and IL-16, trigger the cell death by pyroptosis. So one of the novelty of our study is the induction of pyroptosis by zinc oxide in skin cell has never been reported before. And regarding the autophagy dysfunction triggered by zinc oxide nanoparticle, I think it's well defined. Zinc oxide nanoparticle can trigger both the autophagosome formation and the lysosome dysfunction, and both contribute to the autophagy dysfunction. And the exosome, I think the exosome probably can serve as a bridge or crosstalk between the inflammasome and autophagy. And both the autophagy and exosome release are responsible for eliminating unwanted protein. And the inflammasome can also serve as regulators of protein secretion by exosome and to alert and guide the neighboring cells. So I will go through our experimental parts very quickly. First of all, we provide, again, sufficient physical chemical property of zinc oxides by using the TEM, DLS, and NTA as usual. And then we determine the morphology and viability change of skin cell treated with zinc oxide and UVB. So you can see the zinc oxide can decrease the viability dramatically. And the TEM results indicate the silver can trigger the autophagosome formation and also induce the mitochondrial structural change. And to further confirm the mitochondrial damage, we apply the mitotracker dip to detect the loss of membrane potential, apply the mitosux to determine the mitochondria, the RS generation, and apply the mitotracker green to determine the disrupt mitochondria, the morphology. And the zinc oxide and UV can also induce RS generation and inflammasome activation by using the flow cytometry and some of the protein markers of the inflammasome, such as the NLRP3, caspase-1, and ASC. So zinc oxide UVB can also trigger the pyroptosis in CACA cells, which has never been reported before. And we use the adenosine-5 and PI double staining and use the protein marker of caspase-4, 5, GSD-MD to confirm the induction of pyroptosis. So in addition to our in vitro cell culture model, we also prove the zinc oxide and UVB can induce skin inflammation, inflammasome, and pyroptosis activation in mice. So as shown in here, the thermal, the swelling, and the inflammatory cell infiltration can be found. We can also find the increased protein marker of the inflammasome and pyroptosis. So the zinc oxide induced autophagy dysfunction, again, by using our technique before in skin cells. You can see the AO staining and also the Western blotting of the protein marker and the double immunostaining of the anti-LC3 antibody and anti-LMP1. And all of these results confirm the induction of the autophagy dysfunction. And I think the most interesting finding is that zinc oxide and UVB can induce so-called cell-to-cell transmission of the inflammasome components, which can be mediated by exosome. And we isolate and characterize the exosomes by using some of the important protein marker, using the TM imaging to confirm the cap-shot morphology, and by using the NTA analysis to confirm the size distribution range. And interesting finding is the isolated exosome also contain proteins related to inflammasome and pyroptosis. And we summarize our part two study in this figure. So you can see the ROS always play important roles. The ROS can trigger the mitochondrial damage and then activate the inflammasome and leading to the cell death by apoptosis. The autophagy dysfunction can also be induced, and the autophagy dysfunction can further enhance the secretion and propagation of the exosome to the neighboring poor cells and making the neighboring cells inflammasome activation and also die through the apoptosis. And in the last part of my talk, I asked one question. Could autophagy be an early biomarker of nanomaterial-induced toxicity? And I think the answer is yes, but we still have to provide more scientific evidence to address these questions. And in a recent report, they also demonstrated a systemic exploration of the nano-bio interaction in cell culture model using the autophagy as a selection marker, which can be applied to explore the new products of different surface multiplication of multiple carbon nanotubes. You can see in here the heat map of autophagy induction in the neuron cells, and the higher induction of autophagy indicated a higher risk. And another recent report indicate the mitophagy, which is a selective autophagy of damaged mitochondria, could also have to serve as a potential sensitive biomarker for safety assessments. They also demonstrated that PINK1 immunofluorescence-based high content analysis could be an early, sensitive, and reliable approach to evaluate the impact of nanoparticle. And the sensitivity is even better than the detection of cell viability and the mitochondrial damage. And in our recent published review article, we are trying to propose a predictive and tiered toxicology testing strategy for nanomaterial HELA testing. So you can see autophagy probably can serve as a central important role because the autophagy is highly related to oxidative stress, related to lysosome disruption, and also highly related to the inflammasome activation and mitochondrial damage. So we proposed autophagy could be an early biomarker in the so-called tier 1 and tier 2, the in vitro screening. And the autophagy could also have the potential to serve as the biomarker of the tier 3, the in vivo screening, such as the zebrafish embryos for better align the 3RS principle. And regarding the zebrafish, the embryo toxicity test for nanotoxicity assessment, a recent review article indicate that the silver nanoparticle and the zinc oxide nanoparticle both are among the most interesting research target among all the organic or inorganic nanoparticles. How the underlying mechanisms of embryo toxicity include ROS generation, oxidative stress, DNA damage, lipid proliferation, and so on. However, the autophagy induction in zebrafish exposed to nanoparticles and seldom or never been reported before. So how to develop and establish a reliable nanomaterial trigger autophagy detection system in zebrafish, I think is still a big challenge. And about 2 years ago, we published our first study regarding the zebrafish embryo with the title, The Effects of Choriones on Acute Toxicity of Silver in the Embryos. So at that time, we encountered big problems. When we exposed the embryo to silver nanoparticle, we found that most of the metals accumulate on the surface of the chorion, and only a small part of the nanoparticle can penetrate and get into the embryos. So because chorion is an efficient barrier against the nanoparticle, so I think the zebrafish embryo probably is not a good idea or a good model for comparing the toxicity of different nanomaterials. And right after our publication in this issue, the ISO released a document related to nanotechnology, and they suggest assessment of the nanomaterial toxicity should use the dechlorinated zebrafish embryo. The reason, one of the reasons is because the chorion serves as an exposure barrier. So for some chemicals or nanomaterials, and right now it is not possible to predict which nanomaterials might be blocked by the chorion. So using the dechlorinated embryos for toxicity assessment may not provide direct ecological information, but may help to better identify the potential hazardous nanomaterials. And we totally agree with this viewpoint, and we will apply the dechlorinated embryos in our future study for comparing the toxicity level of the different nanomaterials. Although the zebrafish embryo model is not good enough for comparing the nanoparticle, but I think zebrafish embryo could still be a good model for the study of ecotoxicology. So we recently found that when we expose the zebrafish embryo to natural water, spike it with the silver or zinc oxide nanoparticle, we found the survival rate of embryo decreased, the hatching rate of embryo decreased, whereas the oxidative stress increased and the apoptosis level increased. The autophagy is kind of question because we can find the increase of lysosomal activity, but it is not a direct evidence for autophagy. It's just an indicator of autophagy. And more recently, we have one step forward to achieve our goal because we recently, by using a transgenic zebrafish 9 that can express the green fluorescence in liver cell, and then we found the zinc oxide nanoparticle can cause the induction of autophagy in zebrafish liver by using the immunofluorescence of the LC3, and also by using the messenger RNA expression of the LC3 and ATG5, which is both the important marker and also direct evidence of autophagy. However, there's still a long road to go to establish a reliable nanomaterial triggered autophagy detection system in zebrafish. And I summary some of the important points in part 3 and also give some future perspectives. Firstly, I think it's necessary to consider the use of alternative testing strategies, reduce, to refine, or replace the use of animal for assessing the toxicity of nanomaterials. And we suggest a tiered testing strategy by using the cell lines followed by animals such as zebrafish for improved screening of nanomaterial safety. Because the embryo of the chorion of the embryo chorion is still concerned. So in the future, we should use the dechlorinated embryo for our future study, and develop an autophagy-related transgenic zebrafish probably is an ideal strategy for predicting the possible toxicology or ecotoxicology can be applied in the future for risk assessment. And finally, the more profound understanding of the toxicity mechanisms will also facilitate the development of prevention and the intervention policy against the adverse outcome induced by nanomaterials. Well, that's all I have, and thank you so much for your time, and I appreciate. I want to thank my colleagues Chen Hongzhen, Li Youxuan, Wang Bozhi, Zhou Weiwen, Zheng Fengyu, Chen Chunwan, Yan Xianzhang, and thanks my students, lots of students. Since their contribution, we all name, there's no any possibility to achieve so many scientific findings. And also thanks the finding resources from the government, the Ministry of Science and Technology, Toxic and Chemical Substance Bureau in EPA, Taiwan, and also the Institute of Labor, Occupational Safety and Healthy Ministry of the Labor. And thanks again. Thank you very much. Thank you very much. That's all.
Thank you so much for a very comprehensive talk about nanomaterial safety. Is there any question from the floor, please?
Professor Cano, may I have a question?
Yes, sure, please.
Okay. So Professor Wang, so deep and valuable work. I'm Rishi Huang. I have one question. That is, we all know that zinc oxide has a specific property that it can be decomposed into zinc ion by UV or light, which results in its phototoxicity. So, according to your research, I would like to know how to confirm it is the zinc oxide triggering the pyroptosis, or maybe the zinc ion triggers the pyroptosis. So, thank you. This is my question.
Yeah, yes, I think it's a very, very popular question from the audience when I give a talk such as neurotoxicity. Sometimes the toxicity contribute from the ions, not only the zinc oxide ion, but also zinc ion, but also the silver ion. Always a debate because we believe that toxicity is measured from the nanoparticle because, as I just mentioned, there's a mechanism, we call it the Trojan horse, because when the nanoparticle are taken by the cells, it can be, transfer or fusion with the lysosome, where the ions can be released quickly. So I think the toxicity from the nanoparticle inside the cell is the major contribute. However, some of the study also show that when you expose the cell from the nanoparticle outside the cell, I mean in the medium, they also have some part of ions can be released, right? But I think the level of the ion outside the cell is not toxic enough. It already have some evidence, either in our lab or by other groups. So we cannot exclude the contribution of the ion in when they release from the nanoparticle. But I think the ion play a more important role inside the cell. So after it had be taken up inside the cell, I think inside the cell the ion is more important than outside the cell.
Thank you. Any other question? Yep. Any question? I have a few questions to you, but maybe.
Okay, go ahead.
Yeah. Okay. So yeah, I was surprised that NIH 3T3 cells had such a appetite to eat up the particles, and I had a question. Partly you already answered, but what cell type determines what particle to eat up? I mean, phagocytize. Is there any rule or some surface receptor like a toll-like or something like that?
I think it's a very good question because when a particle taken up by the cell depends on many factors. One of the factors is the so-called protein corona. So inside the body in the circulation system, the surface of the particle can absorb the protein in the environment. And it's a kind of kinetic process. Some different kind of protein come and sometimes go and something like that. So what kind of protein located on the surface of the nanoparticle probably can also decided what kind of cell easy to go penetrate or not. But the detail, I still have good answer for this question.
Okay. If there is no other question, I can proceed with my second question. May I then? About the skin study you did with the zinc oxide and ultraviolet, I think I heard something recently that there is a mechanism that zinc oxide has a immunosuppressive effect so that it heals the skin disease. But your mouse system was a nude mouse?
It's a hairless mice.
A hairless. So is there any
immune system is good.
good? So it doesn't do with this dendritic cells of the skin.
I think it's a normal cell, but only they don't have the hair.
Oh, really? Okay. So I thought zinc oxide might protect the UV damage.
Yes, sometimes.
Okay. Sometimes.
Yeah, sometimes.
Oh, really? Okay. And the yeah, because I'm also running a nano study with Dr. Takahashi in the same institute, and mostly in vivo inhalation study, so that the target is the lung, and we are always thinking about pancreas. No. I mean the macrophage. But do you have some idea about the macrophage and your autophagy biomarkers?
Yeah, we do. Several years ago, we found that some kind of nanoparticle, polystyrene. I mean, the polystyrene can induce autophagy cell death in macrophage, and it depends on the charge. So only the positive charge of the polystyrene can induce the cytotoxicity in macrophage and cell death by apoptosis. But the central or negative charge of polystyrene are no such kind of effects.
Okay. Any other question from the floor? No.
Professor Wang.
Yes, please.
I just remember I have still a question. Did you do the cell transformation of zinc oxide previously? The cell transformation? How about its ability for the cell transformation in normal non-cells?
I don't know what exactly is your question. The cell transfer. You mean the cell line I used is a normal cell or is a transformed cell? Or
it's a normal cell. It's a normal non-cell because most of nanoparticles will come into our respiratory system. So did you do the cell transformation before?
So you mean
it's the carcinogenesis?
Yeah, I know that. I know that. So you asked one question: if it is possible that the nanoparticle can transform the normal cell into the cancer cell, right?
Yes.
I don't really know about this question because some of the area in the nanotoxicology is difficult to study. One of them is the carcinogenicity of nanoparticle, and the other one probably is the kinetic of the nanoparticle. So I think we don't have so many evidence that the nanoparticle can induce the carcinogenicity, even the cell model or the animal model. So probably you can try it in the future, but it is so difficult to do that because nanoparticle, we still don't have much deep information regarding their their fate or their interaction inside the body or inside the cell or in the circulation system.
Okay, thank you.
Okay, thank you so much. Last comment I have as a pathologist is that I was shocked by the acute hemorrhagic pancreatitis you had, and I totally understand that there are so many things we have to research. Thank you so much for your very excellent talk. I had a lot of information from your slides. Thank you. And I'd
Thank
like to close your this session and get back to Dr. Lee. Yeah.
Thank you. Thanks, Dr. Kano and Dr. Wang. This afternoon's session, although we have only 4 speakers, but it actually covered a variety of toxicology aspects, starting from Dr. Sato. You can see that although the ASIATOX only established about 20 or 30 years ago, but there are a lot of progress we have. So now we have 8 societies in our society, ASIATOX society. So may I have the honor to ask the societies who have not have their names and songs to show to audience yet. For example, as I mentioned earlier, that people from CST, Dr. Zhou Pingkun, are you still there? Dr. Zhou Pingkun, of president of CST, or yes, would you like to comment? Speak some words.
Okay. Yes. So it's wonderful webinar, and right. We have so everybody enjoy. I think everybody have enjoyed these seminars, and they're wonderful for speakers, and from the history, ASIATOX, also frontier knowledge in the different area of the toxicologist. I suppose this is a very important platform for our toxicology community, especially at this difficult time, COVID-19.
It seems we have some lag.
Here I would like to also express our thanks on the side of CST and for Professor Lee and your grateful work. Thank you.
Thank you. Thank you, Dr. Zhou. And also Dr. Fu, the immediate past president of ASIATOX. Dr. Fu Lijie, would you like to comment? I cannot hear you. Probably you have to turn on the microphone. No, I cannot hear you. Okay, we just wave our hands and wish you well. And
Hello.
Yes.
Hello. Yeah.
Yes. Would you like to speak a few words?
Thank you.
That's all? All right. And
we can hear you now.
Yeah. So would you like to say a few words? Well, I probably then we also have friends from Malaysia. I think Dr. Rozani Abdullah is still around. Are you still with us?
Hello, everyone.
Yes, yes.
Can I say something now?
Yeah, surely.
Yeah. Hi, everyone. Assalamualaikum. A very good afternoon. I'm Rosaini from Malaysia. I'm actually a new president of the society from Malaysia. And first of all, I would like to say thank you so much, especially to Professor Sato, because I think he's the one who encouraged us to have our own society. And our society is actually already 10 years. 12 years this year. So we are still new and we are still a lot of things to learn from other member society. And I think this is a great opportunity for us to learn together and also to share our expertise in this field. And we know that toxicology is a very niche area. And as a country that are still, we don't have many expert in the field yet. But of course, we do have some, especially our founder for this society, Professor Salman Hussein, that really contributes a lot to our society, and is also an active member until now. But I think this is a great opportunity for us to learn together and also to learn from the other member society in this Asian region. Thank you so much for organizing this seminar, and I think we look forward to contribute more in the next future seminar. Thank you.
Thank you, Dr. Abdullah. And we also have people from Toxicology Society Singapore, that Pram. I saw his name early on, but I'm not sure if he's still around with us. Or some people from Singapore? Maybe not. And we surely know people from Thailand. Dr. Songsak, we mentioned earlier. Would you like to comment and speak a few words? I cannot hear you. Probably you did not turn on your microphone. Yes. I cannot hear you.
Okay now?
Yeah.
Okay. Yes. Well, it is a wonderful afternoon. I think it is first of a kind in ASIATOX that we have this kind of webinars. And also we have to thanks to the Professor Lee that initiate this webinars. And we have this kind of webinar because of the pandemic of COVID-19. So it is a very good start for the organization in this kind of webinar. And I believe that in the future we can continue on before we go to the big ASIATOX next year. In that case, I hope that we can have the on-site in Taiwan next year. I believe that the COVID-19 is stabilized now. In Thailand, we start to open up the country.
Yes.
Traveler come to Thailand not have difficulty in coming in. So anyhow, the webinar today is wonderful. I learned a lot from each speakers that give the very in-depth of the field of expert of each speakers. And especially, I have to thank Professor Sato that initiate the ASIATOX. remember that before the ASIATOX initiated, he invited me to go to Shiba University and talk. At that time, I went to the conference in Osaka, I believe. Mm-hmm. So that is the starting point that ASIATOX formed formally until we finished the formation of the ASIATOX in Hokkaido, I think. Is that right, Professor Sato?
Yes.
Yeah.
Yes.
We have a wonderful time in Hokkaido at that time. Mm-hmm. Yeah. Mm-hmm. And again, I have to thanks to the Professor Lee that initiate this kind of seminar, and thanks to the Taiwan Society of Toxicology that organized the seminar wonderfully. Yeah, thank you very much.
Thank you, Dr. Songsak. Because of the initiation by Dr. Sato, we have the talk on the past and future perspective on ASIATOX. And that's why we have the preliminary discussion. We are supposed to have a closer link to monitor and to have a warning system for the potential toxicants that exist in our environment, especially in Asian regions. So that will be the topic we are going to discuss sometime next year during the ASIATOX 10 in Tainan. So we like to thank Dr. Sato and Dr. Songsak for the very useful information and initiates. Yes, Dr. Sato, would you like to say something?
Yes. I have a few words. First, I'm very happy to meet all the friends. Thai, China, Singapore,
yes,
Malaysia, yes, and also Taiwan. And on behalf of the Japanese speakers, I sincerely appreciate TSTA friends to invite me to this wonderful seminar. Also, I appreciate KMU alumni to have this opportunity. Mm-hmm. And finally, I'm hoping ASIATOX 10 will be fruitful next year. Mm-hmm. Thank you very much.
Thank you, Senator Dr. Sato. I forgot to mention one of our friends, although we said there are 6 society attended this webinar, we also have a friend from Iran. So I'm not sure if our Iranian friend is still around. Would you like to say something to our friends? So
thank you very much for the webinar.
Yes.
Can you hear me?
Yes, loud and clear.
Thank you very much for the webinar. I enjoyed comprehensive talks from colleagues from various parts of ASIATOX, and that was a good experience for myself to attend this webinar. I will share my experience from this webinar with my colleagues at Iran Talks, and I hope we could participate by having presenters from Iran Talks to next series of ASIATOX webinars. So I appreciate it, and I would like to congratulate you for the productive and successful webinar that we experienced today. Thank you.
Thank you. I do hope the success of this first online seminar will bring the continuing of our second and third and subsequent webinar in the future. So I formally invite all of you to continue to contribute to our society, and we are going to have more webinar in the future. So keep in touch, and especially to the friends from Japan, because for such a short time, 3 Japanese friends, Dr. Sato, Dr. Kano, and Dr. Ogra, they when I ask them if they can help, they say, well, why not? And they just help. Within just 1.5 months, then we have this webinar. That's very wonderful. And I would say that's a miracle, actually. And the second, from the first talk, we are going to have the second and so on and so forth. I do hope our link will be going stronger, and we are going to have more closely linking society. And I'll see you, all of you, in the future. Thank you all.
Thank you.
Thank you, everyone.
Thank you.
Bye-bye.
Okay.
Thank you. Bye-bye.
Bye-bye.
Bye.
Thank you. Bye-bye.
Thank you. Good job.
Have a nice weekend. Bye-bye.
Thanks.
Bye. Yeah. Okay.
Bye-bye,
Bye.
Joe.
Bye-bye.
Bye-bye, Joe.
Hi, Lichi.
Hey,
Hey, how are you?
good. How are you?
Pretty good.
Yeah, good. Nice seeing you.
Nice seeing you.
Finally, my voice comes up, right?
Yes.
Excellent job. Good job. So nice. So glad to see all of you. Take care. Have a good summer.
Yes.
Bye-bye.
Bye-bye.
Rain a lot.
Bye-bye. Yeah.
Bye-bye. See you later.
See you.
Yeah.
See you.
See you next time.
See you.